السلام عليكم. أنا طالب ركان حداد من كلية الطب في الجامعة الأردنية. Today we're going to be studying the fourth lecture of Neoplasia. I really want you to know that this lecture is going to be easier than the previous lectures. It's really small. We kind of took everything before, so it will not be this difficult. In addition to that, it's kind of common sense. يعني we're going to realize that this lecture is common sense. يعني كله منطق بمنطق. The hallmarks of cancer. أول شيء شو يعني hallmarks؟ يعني أشياء بتصير وبتخليني أميز إنه هذا الإشي هو cancer. Okay? فبدي إياكم تنتبهوا. Whenever we notice these things, most likely the injury or the type of injury is going to be cancer, or the type of pathology is going to be cancer. Okay. أول شيء the self-sufficiency in growth signals. شو يعني؟ يعني إنه نفس الخلية بلشت تصنع growth signals بكمية كتير كبيرة. The cell itself is starting to create lots of growth signals, and these growth signals are stimulating maybe the same cell, maybe other different cells يعني next to it يعني. Okay, the whole point here is that the amount of growth signals is going to be higher, greater than usual, and more long lasting. Okay, طيب. Second of all حيكون في insensitivity to growth inhibitory signals. كتير منطقي الموضوع. أول إشي بيحكي لك the number of them أو حيصير في self sufficiency، حيصير في growth signals بكمية كتير كافية. Second of all, insensitivity to growth inhibitory signals. And this makes sense because if I have lots of growth signals in a normal cell, there will be inhibitory signals. I will stop them. لكن in cancerous cells, I'm going to inhibit there's going to be insensitivity to the inhibitory signals. Okay, so basically the growth signals are always going to be active. تمام. إذا أول نقطة self sufficiency in growth signals حيكون في كمية كافية من growth signals. تاني واحدة إنه في أشياء بتعمل inhibition. الcancer, it has insensitivity towards it. Okay, the cancer cells are insensitive towards the inhibitory signals, so growth is going to always be active. Okay, third of all, the altered cellular metabolism. This makes sense because the cell is going to start changing. If it was a normal cell, it would have had normal metabolism, synthesized normal stuff in the body, wouldn't have been this bad. لكن because the cellular because the cellular metabolism has been altered or changed, the whole activity of the cell is going to change, and this is going to cause the cancerous or the bad effects, let's say. Okay. Fourth of all, we have the evasion of apoptosis. يعني زي ما حكينا كل شيء منطقي وكل شيء فعليا ماخذينه من قبل يعني. The fourth point, evasion of apoptosis. What is apoptosis? اللي هي cell suicide. Okay, clean cell suicide. Apoptosis, the cell takes itself and dies بكل بساطة. Okay, في extrinsic pathway, في intrinsic pathway, we learned all of that. هاي هي الapoptosis. طب شو بتهمني أنا الapoptosis؟ apoptosis, the cell uses it to get rid of itself if it's damaged. لكن when there's a cancer cell, this cancer cell is going to evade apoptosis, so the damaged cell is going to stay alive. Okay? برضو, limitless replicative potential. We know that cancer cells can replicate in a limitless amount. They can keep on replicating and replicating without caring about the density of cells around it. يعني usually when we have a skin injury, what happens is the skin is gonna cover the wound يعني بكل بساطة. Okay, the cells are gonna proliferate until the wound is covered. بس no more proliferation occurs. The skin ما بيبلش يطلع عن حده، ما بيبلش يطلع عن برا جسمك، لا بضل بمحله. Okay. لكن in cancer cells, that's why tumors form. They're gonna grow without caring about the density of cells around it. طيب. Number 6, sustained angiogenesis. شو يعني angiogenesis? Angiogenesis يعني I'm basically forming مثلا هذا blood vessel وهذا blood vessel تاني. Okay, what's happening now is I'm forming new blood vessels and I'm forming new routes, and these routes are going to start feeding the tumor. Let's say that this circle is a tumor and this is the new blood vessel. Okay, هلا زمان this tumor used to be fed by this blood route and by this blood route مثلا. Okay, تمام. شو يعني it used to be fed؟ يعني it takes nutrients, it takes oxygen, it takes oxygen, it takes other sorts of nutrients like any sorts of macromolecules in the body. It starts feeding on them, maybe sugars, maybe lots of things يعني. Okay, مش بالضرورة oxygen يعني anything. المهم إنه the tumor is going to feed on the things in our body, especially glucose, yani, especially sugars, okay? What's happening now by angiogenesis is I'm providing another route of nutrition towards the tumor cell, okay? So برضه منطقية, it's going to help خلينا نحكي make it even more long lasting. It's going to help it with its life, help it survive, okay? Help the cancer cell survive, which is something really bad, يعني. Invasion and metastasis, this is really obvious. Yani, cancer cells are going to invade tissues and they're going to metastasize to different tissues. And we learned that we have three pathways: seeding, lymphatic pathway, and the hematogenous pathway. Okay. Evasion of immune surveillance. Cancer cells are really tricky. بيجوا بيغطوا حالهم بيفكروا حاله يعني بيخلوا الجسم يفكر حاله انه مش عم بيتعامل مع شيء غريب. Okay? They are really tricky because they act as if they're one of our body cells, which is really bad because our own immune cells are not going to be able to recognize them. If we do not recognize them, then بكل بساطة we won't be able to destroy them. So it's going to keep on harming the body without knowing what's actually going on. Okay? Here we're just, it's just a simple diagram talking about how accumulated mutations are going to eventually cause a really bad cancer. Okay, this is the initiation of the mutation. So we have a normal cell. It's gonna be mutated by a carcinogen. تحول initiated precursor with stem cell-like properties. Now this is an initiated precursor, which means it's ready to become a cancerous cell. Maybe it can return to its original to its original form. لكن most likely no, because you're going to start having cancer hallmarks here, okay? And it has stem cell-like properties, survival موجود, growth موجود, okay? Then it's going to have more mutations happening to it, driver mutations, they're going to affect it directly, okay? They're going to affect it, has عندك founding cancer cell. More mutations are going to happen. حصير في شيء اسمه subclones. We know that cancer is clonal. We talked about that multiple times. شو يعني clonal? It can take the identity of any tissue it wants, no matter what. So it starts with a specific tissue, monoclonal. Then it starts getting a heterogeneous, it starts becoming a heterogeneous substance, okay, or a molecule, whatever you want to call it, okay? With more accumulation of mutations and the emergence of subclones will have a genetically heterogeneous cancer. Now, ممكن تشوفوا انه هاي الحلقة اللي بصور مسلسل انه هاي الليكتشر طويلة. ليش طويلة؟ لانه عم بكرر كتير عشان خلاص واحد ما يضطر يراجع الحصة بعدين. خلاص you just watch it and you're good to go. Okay, قد ما بعيد you're not gonna have any problems hopefully. Self-sufficiency طبعا we already talked about that. يصير في gain of function mutations. we must understand احنا حكينا بالمحاضرة الماضية we mentioned how proto-oncogenes have proteins. When mutations happen to proto-oncogenes, the proteins get activated. Usually the proteins either get activated or inactivated. لكن because it happened to a proto-oncogene, it gets activated. So عندك شيء اسمه oncogene. تمام؟ oncogenes, they encode proteins called oncoproteins. Okay? These oncoproteins, they can be receptors, they can be internal proteins, whatever they will promote cell growth even in the absence of normal growth promoting signals. So they're really bad. شو الفكرة؟ بصير في a gain of function. من شو إجا gain of function؟ من mutation. Mutation happened لمين طيب؟ للبروتو-oncogen اللي كان فيها normal protein. صار البروتو-oncogen تحول إلى turned into an oncogene. والoncogene has an oncoprotein. oncoprotein cancerous بكل بساطة. طيب مالهم هذول؟ The oncoproteins, they promote cell growth. Even if the cell itself or even if there are other signals around it telling it to stop, it will not care. It's going to keep on growing. طيب هون نشوف steps of normal cell proliferation. I'm going to read throughout them really quickly because we already know them I'm going to try to demonstrate it as much as I can. Okay? There are some specific points that we're going to focus at طبعا هاي السيل, this is the nucleus. هاي عندك الريسبتور, يكون receptor tyrosine kinase, okay, which is really لازم نحفظ receptor tyrosine kinase. It has an intrinsic tyrosine kinase. شو يعني tyrosine kinase? By the way, when we say tyrosine kinase, it means that there is a part of the receptor that has an enzymatic ability. شو الانزيماتيك ability? Adding a phosphate group. Limine, the tyrosine, that's it. So basically it's gonna look for tyrosine residues, okay? This is gonna phosphorylate them, kinase بس, okay? اوكي راحت الرسمة نايس عشان اتفلسف طيب anyways بيجي الجروث فاكتور dimerization occurs whatever we already studied that يعني the idea is the growth factor comes and activates this receptor okay the receptor has and خلينا نحكي it has an intermembrane شو اسمه it has يعني we know that it's a transmembrane receptor يعني okay إلها cytoplasmic domain اللي هو من جوا okay this cytoplasmic domain وإلها extracellular domain طبعا here ligand binding happens اللي هي growth factor okay this is a growth factor anyways cytoplasmic or cytosolic domain whatever you want to call it يعني إيش بصير further signals are sent down the growth factor itself it doesn't go in it just binds up top okay بعدين شو بصير second messengers signaling cascades they start entering until they reach the nucleus بيدخلوا جوا they enter the nucleus they affect specific nuclear factories they cause the growth and replication of the DNA and of the cell and whatever okay so هلا نقراهم على السريع binding of a growth factor to the receptor ركز transient and limited activation of the growth factor receptor okay it's transient it's limited which in turn activates several signal transducing proteins on the inner leaflet of the plasma زي ما هلا حكينا okay يعني this transient and limited by the way transient means للحظي يعني بس لفترة كتير صغيرة okay transient and limited ليش limited و transient because if the growth factor stays on its place it's going to cause overstimulation تعمل overgrowth يصير cancer here we're studying normal proliferation okay anyways 3 transmission of the transduced signal across the cytosol to the nucleus by حكينا إما second messengers أو cascade signal transduction molecules شو ما كان okay طيب induction and activation of nuclear regulatory factors they initiate and regulate DNA transcription and the biosynthesis of other cellular components that are needed for cell division such as organelles, membrane components and ribosomes. Entry and progression of the cell into the cell cycle resulting ultimately in cell division. We already talked about that. حلوين. هلا the growth factors. Let's study more about them. We're going to learn about the receptors as well. Cancers may secrete their own growth factors. حكينا they have the ability to secrete them, to create them, or induce stromal cells to produce growth factors in the tumor's microenvironment. يعني it can tell the tissues around it okay يعني near to it proximal to it whatever to start synthesizing growth factors for it الكانسر هون acts as a leader هذا الليدر بده يستغل كل حدا حواليه بيروح بيحكي للتيشوز اللي حواليه أعطوني كل growth factors اللي عندكم okay so basically he induces the stromal cells around it to produce these growth factors in the tumor's microenvironment طيب Most soluble growth factors are made by one cell type and act on a neighboring cell. شو يعني إنه في خلية or there's a cell secreting a growth factor and it's affecting a nearby cell. This is called paracrine stimulation. Okay, paracrine action. إنه one cell type is secreting a growth factor. Okay, and a neighboring cell is taking it or is using it for proliferation. Hence, we have paracrine action. طبعا some cancer cells they have the self-sufficiency by acquiring the ability to synthesize the same growth factors يعني instead of depending هاي cancer cell this is cancer okay instead of depending on another cell to synthesize its growth factors which might not be really good شو بتعمل it starts synthesizing its own growth factors okay it won't depend on any other cell okay obviously زي ما حكينا it can use other cells around it نعم بس وين تصير مشكلة لسه أكبر when they start synthesizing their own growth factors. Okay. Many glioblastomas secrete platelet derived طبعا growth factors are many types في platelet derived في whatever يعني مليان أنواع يعني هي بحطوها هيك زي حرف أو حرفين قبل GF. GF here is growth factor. Okay. So many glioblastomas secrete platelet derived growth factors and express the PDGF receptor. Obviously إذا أنت you're gonna synthesize platelet-derived growth factor for yourself, you're going to need a platelet-derived growth factor receptor. You need to receive it by its own receptor, okay? And كل growth factor, each growth factor has its own receptor, so please be careful to that. طيب. Many sarcomas make both transforming growth factor, TGF alpha, and its receptor. يعني برضه we must memorize إنه glioblastomas they secrete PDGFs and they have PDGF receptor. طيب. What about specific many sarcomas? What do they have? They have TGF, transforming growth factor alpha. طب غير transforming growth factor alpha, they have its receptor. Okay? طيب. In some cases, the tumor cells will send signals to activate normal cells in the supporting stroma to produce growth factors that promote the tumor growth. We already talked about that here, when they're gonna cause their own environment to produce their growth factors. هذا repetition بسيطة. Okay, growth factor receptors حكينا من الريسبتور هذول هم الريسبتور تايروسين كاينيز. They have an intrinsic tyrosine kinase activity. أكيد مش مطلوب مننا نعرف كل سلافتها والسيجنالينج وإنه إيش بصير. خلاص في أشياء بسيطة هيك يعني أبسط بكتير ممكن ناخذ السيتولوجي. Okay, it's activated by the growth factors binding, while others signal by stimulating the activity of downstream proteins. حلو. إذا some growth factors بيعتمدوا أو they depend on a receptor tyrosine kinase. Okay, how is it activated? When the growth factor binds. Okay, and other growth factor receptors they just act or they start signaling by just stimulating the activity of downstream proteins. Honestly, both of them are gonna يعني I believe that this sentence is kind of inaccurate. No, just both of them are gonna do the same thing. They're gonna by activating the receptor, you're gonna activate proteins on the inside of the cell. يعني لا حول ولا. Many of the growth factor receptors نفسهم they can be oncoproteins. Okay, so we must understand ايش هم حكينا الoncoproteins. They induce the cancerous cell growth, مش cellular growth, the cancer cell growth. Okay, تمام. طيب إذا what about the receptors? Can they act as oncoproteins? أكيد they can. Why can they act as oncoproteins? Because they are going to be able to receive growth factors. If it was a normal receptor, maybe it will stop the effect of the growth factor. It will have enough of it that will just cause the growth factor amount synthesis to stop. Okay? Like when it gets mutated, it turns into an oncoprotein, it will not sense anything. This is what we mean by the inhibitory of the growth factor's sensitivity. Insensitivity to the growth factor inhibition. Okay? This is really important to start linking the hallmarks to these. تمام. Examples on some receptors, the epidermal growth factor اللي بيستقبل شو؟ epidermal growth factor ده شيء صراحة. So the epidermal growth factor receptor, what does it accept? The epidermal growth factor. Okay, ERBB1, just some statistics يعني مش like just some خلينا نحكي things that we need to memorize يعني. It's overexpressed in 80% of squamous cell carcinomas. شوف قديش this receptor is how خلينا نحكي how dangerous it is and how it acts as an oncoprotein in 80% of the squamous cell carcinomas. طيب of the lung. 50% or more of glioblastomas, and 80 to 100% of epithelial tumors of the hand and the neck. Yani whenever someone comes into the clinic with an epithelial tumor in his hand or in their hand or on their neck, okay? If you want to know what type of receptors are present on the cancer cell, we would find 80 to 100% ERBB1 is overexpressed. It has too much of it, okay? So, the HER2 or ERBB2 طبعا this is related to breast cancer ovaries cancer as well, is amplified in approximately 20% of breast cancers. Okay, so this can actually act as a خلينا نحكي an idea for therapeutic or خلينا نحكي targeted medicine so we can target a specific receptor and work on it to prevent the cancer or to treat the cancer. And in a smaller fraction of adenocarcinomas of the lung, ovary, stomach, and these tumors are exquisitely sensitive to the mitogenic effects of small amounts of growth factors. Okay, the tumors that happened here, the whatever sort of malignant tumor horn, due to the increased amount of receptors, okay, they're gonna be highly affected and highly sensitive to small amounts of growth factors. الميتogenic effect اللي هي الميتogenic pathway whatever يعني we took in cytology يعني. HER2 is really significant in treatment يعني, as I just mentioned. Tyrosine kinase activity is stimulated by point mutations or small indels that lead to subtle but functionally important changes in protein structure or gene rearrangements that create fusion genes, encoding the chimeric receptors, which we learned, I believe, last lecture or the one before it. No, last lecture. Okay. What am I reading here? شو هذا التخبيص? Okay, the tyrosine kinase activity. هلا بدك تعرف الريسبتور هيو. Let's say this is the cell, this is the nucleus. The receptor is not this big. I'm really sorry for the bad drawing. Okay. What happens is it has an intrinsic tyrosine activity, يعني tyrosine kinase activity. So maybe it will put maybe it will put a phosphate group here and a phosphate group here as a starter يعني. Then the proteins are يعني inside of the cytosol, the signaling proteins are going to start they're going to start binding here. Okay. They're also going to get phosphorylated مثلا. تمام. Okay. هلا if there's an increase in the activity of tyrosine kinase, you might find way more phosphates. I need you to know that each protein, خلينا نحكي إنه كل protein, it has to do with maybe one cellular proliferation, and this is the normal thing. Okay, so here I want 2 cellular proliferations. This is not true at all. This is just an idea for you to understand. But what happens if a mutation occurs? Okay, so now there are 6 binding sites. So instead of the normal 2 binding sites, 2 normal cell proliferations, they're going to happen 6 mutated proliferations. Okay, thus examples in which the tyrosine kinase activity is going to be stimulated by mutations and leukemias, lymphomas, and certain forms of a sarcoma. Okay. طيب نحكي الآن downstream signal transducing proteins. The signaling proteins that couple the growth factor receptors to their nuclear targets. So we know that we have some sort of second messenger activation, second messenger signaling, and specific proteins that can signal as well. The idea is that the first messenger is going to bind on top. It cannot actually enter to the nucleus يعني. لكن its effect is going to run down to the nucleus one way or another by specific proteins. Okay, that we're going to discuss يعني. Okay, we're going to discuss the most important ones يعني. The signals are transmitted to the nucleus through various signal transduction molecules. The 2 important oncoproteins in the category of signaling هم RAS وال ABL. Mind you, they're both normal. They're present normally. That's why we call them proto-oncogenes. Okay, and them themselves they're proto-oncoproteins. Like when a mutation occurs, they become اللي هم oncoproteins and oncogenes. طبعا an oncogene makes the oncoprotein. There's a gene for RAS that creates RAS. Okay, becomes an oncogene. كان proto صار oncogene. Okay, الoncogene instead of synthesizing the normal protein, it's going to synthesize an oncoprotein. Okay, RAS هات خلينا نشوف. It is the most commonly mutated oncogene in human tumors. يعني whenever you want to think about a tumor, first of all, think about mutations. After mutations, what should I think about? The place of the location. Place of the location, habibi. Place of the mutation. The RAS is the most common oncogene to be mutated. Okay, approximately 30% of all human tumors, they contain the mutated RAS genes, and the frequency is even higher in some specific cancers, like pancreatic cancer and adenocarcinoma. It can reach 60, maybe 70%. Of these cancers, they have mutated RAS genes. RAS is a monomeric G protein. We know that this is a monomeric G protein. We know that. We studied it like a million times. يعني بنعرف إنه G proteins can be trimeric or monomeric. Okay. وشو هو الجي بروتين؟ الجي بروتين is a GTP dependent protein. If it wants to be active, it has to be bound to GTP. هيك this is an active G protein. تمام؟ طيب, when can this activity be cancerous? If the GTP is always bound. Okay? If always bound. Okay? طب بالمنطق إذا صارت mutation يعني صار cancer. Okay? يعني صارت mutation للراس obviously صار cancer. Okay, cancer happens. طيب what will it do? I just told you the G protein can be active if GTP is here. طيب if a mutation happens, will GTP ever leave? Or will it ever get dephosphorylated to GDP? You're going to tell me no. So the mutation works on keeping GTP in its place. بيخلي GTP محلها. هون, this is the mutation that we're talking about. Okay? The mutation that did not allow the GTP to get dephosphorylated to GDP. Okay? طبعا there are lots of mutations that can occur. We're going to talk about them now. Okay? So, when are they active? G proteins لما يكون معهم GTP. لكن when are they inactive? لما يصير عليهم GDP. So what happens? You basically just dephosphorylate it. Okay? تخلص من واحدة phosphate. Okay? طيب how can we activate it? هي ال G protein هي عليها GDP. What happens is it's gonna get exchanged. نعم exchanged مش phosphorylated, exchanged. Okay? بتبدل يعني تطلع الجي دي بي وبتدخل الجي تي بي. تمام؟ Really easy. Anyways, normally RAS flips back and forth between excited signal transmitting state and a quiescent state. Quiescent يعني رايقة هادية فيش growth فيش شيء أنا قاعد ما عندي شيء دخل فيه. Okay? And it flips between them بمعنى cells are continuously proliferating, so I want the signal to come and go whenever I need it. فدائما في يعني flipping back and forth between them. Here we talked about that. Stimulation of cells, طبعا RAS is inactive about GDP. Stimulation of cells by growth factors like the epidermal growth factor, platelet derived growth factor. شو بتعمل؟ يعني لما الجروث فاكتور يرتبط، الافكت الاول شو بصير؟ هاي طبعا ارتبط الجروث فاكتور. انا ليش هيك برسمها كانها كروموسوم؟ مش عارف ليش. اوكي بيجي جروث فاكتور بيرتبط. اوكي بيكون في جي بروتين موجود هيك هون. هذا هو الراس اللي بيكون هون بستنى. Okay, what happens is لما الجروث فاكتور يرتبط, when the growth factor binds or the ligand binds يعني هم الجروث factor, whether it's epidermal growth factor or platelet derived growth factor, okay, what happens is the G protein that already has GDP is going to start having GTP. يعني I exchange the GDP for GTP. Now the G protein is activated. Okay. طيب, so now there's a conformational change. This protein has been activated. RAS is going to activate another pathway. RAS activates RAF, which activates MEC, ERF, whatever, MEC, ERK, نسيت يا زلمة, مش مشكلة عادي. هلا بنتذكرهم. يعني عادي بسيطة. Okay, so يعني بكل بساطة the growth factor binds, which causes the exchange of GDP for GTP. Okay. مش نسيت أن ترى خربطت بالأحرف بس يعني anyways, signaling by RAS involves the sequential steps. شو هم؟ خلينا ناخذ شوية معلومات أول. Let's just have some information in mind يعني. The active RAS يعني RAS that has GTP is really short-lived because of the activity of an intrinsic guanosine triphosphate ASE. Okay, guanosine triphosphatase. Okay, شو يعني triphosphatase? There's an intrinsic activity. Intrinsic يعني داخلية, منها وفيها. شو هي هاي الactivity? It changes the GTP to GDP by dephosphorylation. This is called the GTPase. Okay? فبكل بساطة a normal G protein has an intrinsic activity that dephosphorylates GTP into GDP. But what happens if a mutation occurs to the GTPase activity? It will not dephosphorylate GTP and the G protein is always going to be active. تمام? Okay. RAS hydrolyzes GTP to GDP, so it releases a phosphatic group and it returns the protein to the quiescent GDP bound state. Okay, so we must understand that the GTPase activity of the activated RAS is magnified dramatically. شو بتحكي انت؟ by a family of GTPase activating protein gaps. ايه طب صار عندي هيك تخبيص كتير. اوكي نفهم. هي جي بروتين تمام مع عليه جي دي بي. يا حبيبي شو صار؟ ايش شنو هذا؟ لا ما اتفقناش هيك. اوكي هي الجي بروتين عليه جي دي بي. طيب صار اكتف. شو يعني صار اكتف؟ عملنا exchanging للجي دي بي صارت جي تي بي. طيب مجرد ما تصير هاي الاكتيفيتي هل هون صار عندي انا active state. الاكتيف I don't want it for a long time. I really don't. I want it just for a specific amount of time. So what happens طبعا we must understand that there's a built-in GTPase في عندنا GTPase. GTPase returns this to this بكل بساطة. Okay. طيب the GTPase is also present in the G bound to the ADP, which is the inactive state. لكن there are specific proteins called gaps. هذول هم ال GTPase activating proteins. They activate the GTPase and magnify its activity. يعني بيزيدوا الاكتيفيتي تاعتها. طب لما يزيدوا الاكتيفيتي تاعتها شو بصير؟ It's gonna start being able to dephosphorylate the GTP فبتخلص من الاكتيف ستيت وبترجع للانactive state وهذا اللي احنا بدنا اياه يكون في عندنا regulation. Okay? Really easy. So الجابس هم عبارة عن breaks. They prevent uncontrolled activation. How do they do that? They favor the hydrolysis of GTP to GDP. Activated RAS will stimulate the downstream regulators of proliferation by pathways that regulate the growth that converge on the nucleus and alter the expression of genes that regulate growth, such as the MYC gene, a gene that regulates growth. Okay, just بكل بساطة. For example, BRAF, which lies in the so-called RAF-ERK-MAP kinase pathway, is mutated in more than 60% of melanomas and is associated with unregulated cellular proliferation. Okay, so this is just a specific protein. It lies in this pathway, RAF-ERK-MAP pathway. Here RAF-MEK-ERK صح? Is RAF-ERK-MAP kinase pathway. Okay. فشو بصير؟ a mutation happens to the BRAF in more than 60% of melanomas and is associated with unregulated cell proliferation. شو هو فيه؟ unregulated cell proliferation. The cells are going to divide without regulation. تمام؟ RAS most commonly is activated by point mutations. يعني RAS what type of mutations happen to them? هم point mutations, okay, that happen to the amino acid residues that are either within the GTP binding pocket or the enzymatic region that carries out the GTP hydrolysis. شو اللي انت عم تحكيه؟ انت ترى بلشت تأسرع كثير. اسرعت على اللي فوق لانه صراحة سهل. هنا في فكرة. هي الجي بروتين again. Actually هي مثلا الجي بروتين اللي هو الراس. اوكي. هون في لها binding spot. تمام. هون في لها binding spot. Now, if a mutation happens to this binding spot, هاي الاسم هيك mutation. Okay. What will it do? It might lead to the خلينا نحكي خلينا نحكي an increased strength to the binding to GTP. طيب شو برضه ممكن يصير؟ برضه the mutation can occur to the GTPase itself. طيب إذا صارت ل GTPase I will not change GTP to GDP. شرحناها قبل صراحة. Okay. Both kinds of mutations, what they do is they just interfere with the breakdown of GTP, and this is essential to inactivate RAS. Okay, so a mutation does not allow the breakdown of GTP. يعني a mutation does not allow the inactivation of RAS. يعني the mutation causes overactivation of the RAS. RAS is thus going to be trapped in its activated GTP bound form. The cell is forced into continuous proliferation. Activating mutations in RAS can be mimicked by loss of function in gaps. شو هم الجابس؟ GTPase activating proteins. What if they lose their ability? إذا هذول الجابس they lost their ability, they won't be able to activate the GTPase. Okay? شو يعني؟ يعني GTP is always going to be present instead of GDP. Okay? So it will lead to failure to stimulate the hydrolysis and it will trap RAS in the active state. An example, neurofibromatosis type 1. The gap اللي اسمه neurofibromin 1 NF1 is mutated. Okay, يعني this is just an example. In neurofibromatosis type 1 إنه في عندك a gap اللي هو GTPase activating proteins. A mutation happens to it. Okay, الgap اسمه neurofibromin 1. It gets mutated. Okay, which causes neurofibromatosis type 1. طيب كمان example, a tumor suppressor gene, a TSG called P10. This is a negative inhibitor of PI3 kinase. It's frequently mutated in carcinomas and certain leukemias. Okay, so P10, it's a negative inhibitor. بمعنى it's going to calm down the cellular proliferation. But if it gets mutated, it will not be able to do that. Okay? طبعا here we're just يعني اشي على السريع. طبعا look here طبعا ال PI3 kinase بكل بساطة what it does is it induces cell cycle progression يعني it induces cell proliferation. تمام؟ P10 it's a negative inhibitor يعني it calms it down. Okay? خلينا نحكي it causes a block here خلاص. لكن if a mutation happens it proceeds. Anyways هاي طبعا ال receptor ال growth factor receptor. The growth factor binds. شو بصير؟ يصير في activation of the RAS. كيف ال activation؟ I replace GDP. يصير عندك active RAS. طيب هلا الراس هذا شو بيعمل؟ ببلش لك الراف ERK يعني the RAF MEK ERK pathway. Okay. Anyways اللي هي الميتوجين activated protein kinase MAPK. Okay. Which will induce cellular proliferation, which will activate the MYC gene, which activates the MYC protein that activates the cell cycle. أو اللي هي ال PI3 kinase pathway يعني بكل بساطة the activated RAS activates transcription يعني activates cellular proliferation the cell cycle. طيب شو بصير؟ The activated RAS بصير GTP hydrolysis by the GTPase which needs the gaps فبيتحول لل inactive form وخلص العملية كلها بتهدى. اوكي؟ اوكي بالنسبة الآن to the ABL oncoprotein or proto-oncoprotein إلها a tyrosine kinase activity and this activity is negatively regulated by its internal domains. Okay? يعني it's also a protein. This protein has specific domains. Domains يعني a specific active part that has a three-dimensional shape and function. يعني it's برضه a polypeptide part of the ABL. تمام? This part controls the activity of the tyrosine kinase. Okay, so it's regulated by its own self. Okay, it's regulated by its own amino acids خلينا نحكي or by its own polypeptide. It does not depend on مثلا a gap or a GTPase. لا هاي ايش خلص الactivity هون it's highly dependent on the internal domains of it يعني on its own structure. In chronic myeloid leukemia CML in certain acute leukemias, a part of the ABL gene is translocated from its normal site when هذا موجود on chromosome 9. It goes to chromosome 22, forming Philadelphia chromosome, PH chromosome. Discussed that last lecture. Okay? So شو بصير? The ABL gene in normal, in cases of certain acute leukemias and chronic myeloid leukemia, this gene translocates itself يعني بيمسك حاله بيروح from 9 chromosome 9 to 22 forming what's called a Philadelphia chromosome PH chromosome. طيب this causes the fusion of two genes واحد منهم ال ABL because it has been translocated. طب مين الثاني؟ جين اسمه BCR مش PCR BCR. اوكي بصير في عندك فيوجن جين BCR ABL. شو هذا بيعمل؟ هذا له تيروسين كاينيز اكتيفيتي. حلو. طب شو اللي بصير هلا؟ طب عادي ABL has a regulatory site. اوكي يلا بطل عندي ABL صار عندك fusion of BCR and ABL. So when ABL tries to stop the kinase activity, the BCR tells it, no, I'm not used to that. I'm going to keep on activating. Tyrosine kinase is always going to be active. Okay? So the BCR-ABL protein will activate all of the signals and it's a potent stimulator of cell growth. Imatinib mesylate or Gleevec is BCR-ABL kinase inhibitors that has a dramatic clinical response in patients with chronic myeloid leukemia. Okay? So something that is given as a kinase inhibitor to people with chronic myeloid leukemia or acute leukemia, certain acute leukemias يعني. Okay? And it actually has a dramatic clinical response. Okay? It's really important. طيب. Nuclear transcription factors. شو هذول؟ نعرف انه بالنوكليوس في عندك شيء اسمه transcription factors. they are highly responsible of transcription. They regulate transcription. أصلا the whole point of this signal اللي بتيجي من برا, this signal here, the growth factor, the whole point of it is to activate the transcription factors. Let's call them TFs. Okay? Transcription factors, they will basically get on the DNA and start transcribing it. Okay? ببلشوا يحكوا لك بلشوا يقرأوا ال DNA ويعملوا لك whatever they want, proteins for cellular growth, for whatever. So the mutated RAS or ABL, what will it do? It's going to activate the signaling pathway. يعني the nuclear transcription factors are always going to be activated. Okay? So if you continuous stimulation of nuclear transcription factors that drive the expression of growth promoting nuclear genes, we must memorize these genes. They cause growth. The MYC gene, MYB gene, the JUN, the FOS, and the REL oncogenes. كلهم, they are genes that if mutated, they can cause cancer. Okay, they are responsible for the overgrowth. Okay, they're growth promoting genes. Which of the following is a growth promoting gene? هدول. Function as transcription factors that regulate the expression of growth promoting genes such as cyclins. What do? They act as transcription factors. What do? They cause more expression of cyclins, or in general growth promoting genes. يعني نفس the growth promoting genes, what will they do? They will function as transcription factors, okay, activating more growth promoting genes. Okay, so a growth promoter activates another growth promoter until it reaches the final growth promoter, like the cyclins. Okay, the cyclins are really important for the activity of the cell cycle. They're really important. Okay, طيب. MYC is involved most commonly in human tumors. يعني in this lecture, the last lecture, the one before it, we mentioned MYC a lot. يعني. MYC activates several growth-promoting genes, including cyclin-dependent kinases CDKs. الcyclins مستحيل يشتغلوا بدون CDK. الcyclin وفي عندك cyclin-dependent kinases. Okay, these are really important with each other. Okay, so CDKs والcyclins. Okay. cyclin نعرف انه هو السايكلينات cyclin dependent kinases اللي هي الانزيمات الفسفرة المعتمدة على السايكلين. I kind of mistake here. The CDKs depend on the cyclins. يعني okay. The cyclins, they bind to the CDKs and they guide them. Okay, so yeah, the cyclin dependent kinases, they depend on the cyclins. Okay, مش العكس. Okay. الساعة 1 الفجر وعم بصور يعني محاضرة يعني forgive me for any simple mistakes. Examples: MYC translocation from chromosome 8 to 14 in Burkitt's lymphoma. خدناها. A highly aggressive B cell tumor. Burkitt lymphoma B cell tumor. Okay. MYC also is amplified in the breast, colon, lung, and many other cancers. So this shows the importance of MYC gene to cancer. Okay, the more activated MYC gene is, the more cancer can appear. Okay, or يعني خلينا نحكي, we can notice it in many types of cancers. This shows how important it is for cancer. Okay, NMYC gene is amplified in neuroblastoma, just because N neuroblastoma. Okay, LMYC gene in the lung. Okay, so not really that difficult. تمام, هي normal chromosome 9 على ال ABL1 gene شو صار? This is a normal chromosome 22. على ال BCR صار في translocation, صار في fusion gene. هذا it's a tyrosine kinase, has a tyrosine kinase activity that does not stop. Okay, طيب. Cyclins and cyclin dependent kinases, يعني cyclin dependent, بيعتمدوا على cyclin, they rely on the cyclins. Okay, progression of the cells to the cell cycle is controlled. مين بيعملها controlling? اللي هي cyclin dependent kinases مع الcyclins يعني. Okay, so هم actually يعني without the cyclins they wouldn't work. يعني هم they are kinases, they're really needed, and the cyclins will guide them throughout their job. Okay, CDKs, yeah, they're going to be activated by binding to the cyclins. Cyclins are characterized by a cyclic nature. عشان هيك اسمهم cyclins. Okay, cyclins because they're cyclic nature. Offshoot of production and degradation. They have a cyclic nature of production and degradation. The CDK inhibitors, CDKIs, they have negative control over the cell cycle. They inhibit the cyclin-dependent kinases, يعني they inhibit the phosphorylation that is needed for the cell proliferation or the cell cycle to occur. For CDKIs, what do? They basically inhibit the cell cycle. Is this useful? Yes. بوقته it's useful. مش بوقته أبدا. Okay? طيب. Expression of these inhibitors is down-regulated. شو يعني down-regulated? Down-regulated means that it's gonna get lower. بس بكل بساطة. Okay? The amount is gonna get lower. Amount of شو؟ الinhibitors. طب by mean? By the mitogenic signaling pathways. اللي هم the RAF, ERK, MEK, تخبيص, the MAPK pathway. Okay? The MAPK pathways. الفكرة إنه when a growth factor binds, it gives the mitogenic signaling pathway the okay to occur. يعني هاي بتصير متى لما growth factor يرتبط. Will I need inhibitors or activators of the cell cycle? I'll need activators because I want the cell to grow. فهير the CDKIs are going to be إيش؟ Are going to be down regulated. Okay? طيب ليش? So we can promote the progression of the cell cycle. The CDK cyclin complexes, they phosphorylate crucial target proteins, we know that. On completion of this task, cyclin levels decline rapidly. Cyclins of 4 types, يعني. What do? They synthesize themselves and they degrade themselves rapidly. Okay? When they finish their job, they degrade themselves. Okay? So, more than 15 cyclins have been identified. Okay. A and B. These appear sequentially during the cell cycle and bind to one or more CDK. Okay, so just a little that the most important ones are the D, E, A, and B cyclins. Okay, more than 15 cyclins have been identified. The most important ones are D, E, A, and B. They appear sequentially during the cell cycle and bind to one or more CDKs. Okay. Sometimes things can affect the expression of cyclin D or CDK4. This is a common event in neoplastic transformation. Okay, so usually in a neoplastic transformation, cyclin D or CDK4, they are a really common event in neoplastic transformation. The one cyclin D is really important because it starts the whole cell cycle. So if a mutation happens, usually a common event is a mutation happens here, CDK4 and cyclin D. So basically this will cause a neoplastic transformation. The cyclin D genes are overexpressed in many cancers, including those affecting the breast, esophagus, liver, and a subset of lymphomas. So شوف قديش هي موجودة بكثرة بهدول الكانسرز. ليش? Because cyclin D is responsible for the cell cycle. If it's mutated, maybe it's going to cause the hyperactivation of the cell cycle, tumors are going to form. Amplification of the CDK4 gene occurs in melanomas, sarcomas, and glioblastomas. Okay. Mutations affecting cyclin B and cyclin E in other CDKs can also occur, but they're much less frequent than the cyclin D CDK4. While cyclins arouse the CDKs, CDK inhibitors, they silence CDKs and exert negative control over the cell cycle. They inhibit the cell cycle. مين like ال P21 طبعا this is a family اللي هم P21, P27, P57. Okay, so we just have to memorize them, not really difficult. بينما the other family, it has selective effects on mean cyclin D. With that CDK4 and CDK6. Okay, مين هم هدول؟ P15, 16, 18, 19. try to memorize إنه أي إشي فيه طبعا the number 1 okay is gonna be for cyclin D, CDK4 and 6. بينما in general التانيين تاعون that inhibit the CDKs broadly, they have the number 2 or 5 21, 27, 57. Okay, محكي that has the number 1 قصدي أول okay. هدول طبعا P15 16 18 19 we call them the INC4 proteins. Okay, expression of these inhibitors is going to be down regulated. احذر بمين طبعا هذول inhibitors. هلا حكينا هالمعلومه هاي. inhibitors بدك الانهبيتور يصير بالcell cycle. Do we want them while the cell cycle is occurring? No. طيب what starts the cell cycle? Growth factors. Growth factors شو بيعملوا? Mitogenic signaling pathway. So this pathway inhibits the inhibitor. It down regulates the inhibitor. So it will promote the cell cycle. طيب, this is an example. P27, it's a CDK1, inhibits cyclin E. It is expressed throughout G1. مثلا. Mitogenic signals inhibit P27, relieving the inhibition. Okay, and it will allow the cell cycle to proceed. CDKIs are frequently mutated or otherwise silenced in many human malignancies. Examples, germline mutations of P16 are associated with 25% of melanomas. طيب, acquired deletion or inactivation of P16 يعني هذا it's an INC4 is seen in شو؟ طبعا some statistics you have to memorize. Okay, اللي هم deletion or inactivation of the P16 itself. وين بتصير هاي؟ 75% of the pancreatic carcinoma, 40% to 70% of glioblastomas, 50% esophageal cancers, and 20% non-small cell lung carcinomas, soft tissue sarcomas, and bladder cancers. Okay. Good luck, doctors. I hope you enjoyed this lecture. Hopefully. ونختم محاضرة اليوم بحل الأسئلة زي دايما. How do oncogenes contribute to cancer development? طيب. Do they inhibit normal cell growth and promote apoptosis? أكيد لا. Oncogenes promote cell growth and division even without external signals. Okay. أكيد هي صح يعني. Oncogenes lead to production of proteins that suppress immune response. شو دخل? Oncogenes cause cells to become resistant to chemotherapy. Nope. Oncogenes, they promote the cell growth and division even without external signals. Okay? What is meant by self-sufficiency? يعني the cell is able to grow without needing external signals. ليه؟ هي بتصنع اللي عندها. Philadelphia chromosome, it's basically a chromosome that happens when the translocation occurs from chromosome 9 to 22, يعني the ABL gene, and it's relevant to chronic myeloid leukemia. Okay, then translocation بصير the ABL gene. D. What is the relationship between mitogenic signaling and CDKs? Mitogenic signaling, they down-regulate CDK inhibitors, promoting cell cycle progression. خلينا نكرر السؤال مرتين. مش عارف ليه. How does the expression of cyclin D relate to various cancers? طبعا تخربط كان لازم احط سؤال تاني صراحة بس تخربطت وحطيت هذا فلا حول ولا. How does the expression of cyclin D relate to various cancers? Cyclin D is going to be overexpressed in many cancers because it leads to cell cycle. What is the significance of growth factor receptor in cancer? شو بيعمل? Growth factor, they drive proliferation survival. MYC translocation on Burkitt's lymphoma, it basically activates the growth promoting genes leading to aggressive tumors. ABL proto-oncoprotein, how is it involved in leukemia? طيب, ABL, it's a proto-oncoprotein, is a tyrosine kinase involved in leukemia. Okay? Okay. What mutations commonly affect RAS leading to continuous cell proliferation? خلينا نشوف. الRAS بنعرف انه mostly بصير لها point mutations. Okay. Point mutations الRAS. What role do growth factors play? هلا حليناها. How does sustained angiogenesis support tumor growth? شو بتعمل angiogenesis؟ تعطي nutrients and oxygen for the tumor growth. Okay. يعطيكم العافية دكاترة. I hope you enjoyed this lecture.